All right, it's that time again. Hang out with your friendly neighborhood virus man. All right, let's talk coronavirus. So, we've covered how the virus gets in, and very similar to the way HIV got in, very similar to the way some of the other viruses we'll talk about get in. Now we want to talk about what happens once the virus is in. I apologize, the picture up there is not spinning today. I could not find an animated GIF for this. Oh well, yeah. But this is relevant. This is the factory where coronavirus makes all of its new RNA. And the green, sort of spaghetti looking stuff inside, that is the coronavirus RNA. And the little red things, I don't know if you can see them, there are just a couple on here. Those are the pores. So that's how the RNA comes out after it's been made, and it's how raw materials go in. But more about that in a moment. For now, let's talk about the general outline. What's the virus trying to accomplish? So, this is an RNA virus and it has no DNA stage. So, this is RNA only, and it's a single stranded positive sense virus, which means you could translate it and make proteins right away from the genome that comes into the cell. And that's kind of what coronavirus is going to do. So, unlike a retrovirus, which you remember packed enzymes with it, it had protease with it, and integrase and reverse transcriptase all hanging out inside the virion. Coronavirus packs nothing. Nothing but its genes. Yeah. And its genes will then be translated once it gets to the cell, and that will produce the first copies of the polymerase and all the other enzymes that will make this process happen. So yeah, it's a different philosophy. This is the way positive strand viruses do it. They translate what they need when they get there. HIV is behaving like a negative strand virus. Negative strand viruses can't be translated directly, and so they would need to pack with them a polymerase and whatever components they need to actually get the replication process started. And then once it's started, they can always make more proteins of any kind, and they can take over the cell. So our virus is going to bind, it's going to enter, and this is going to be endocytosis, so it's going to be in a little endosome, and it's going to get acidified, and it's going to burst out of the endosome, or rather create a fusion pore and merge with the endosome, releasing the genome into the cytoplasm. Once the genome is out, so that genome is going to be the little g RNA for genomic RNA. And we've got to specify for coronavirus because it has a genome, and then it's going to make several smaller versions of the genome. And those are called subgenomic RNAs. So you'll see those as sgRNA. Or, because this person is apparently a goofball, or listening to goofballs, sgmRNAs. Now, I know this person is actually not a goofball, and I think this is a fantastic review. The link for it is down there. Almost all the slides that we're covering for coronavirus replication are going to come from this particular paper. It is written by the best people in the field, and they're the ones that discovered a lot of the parts of this. So I don't see how you could get a better version. Okay. Once the virus is in, or the virus genome is in the cytoplasm, it will then find its way to a ribosome. We don't know if there's any special way that it does that, but there are sequences in the 5' untranslated region that seem to enhance translation. Like if you put a regular gene with the beginning of a coronavirus, you will make more of that. So it's probably doing something like an IRES. That's an internal ribosome entry site, IRES. But it hasn't been fully worked out as far as I understand. It's going to be translated then, and it's going to produce the first set of proteins that the virus makes. So in this first set, we're going to generate all the enzymes that the cell needs, that the virus needs to take over the cell, and all the enzymes the virus needs to make copies of itself. So it's going to make proteins that shut down parts of the immune response in the cell, and it's going to make proteins that will directly participate in all the various activities that it takes to copy a brand new coronavirus. And there are several, but we'll get to those as well. It's going to do this in one of these. They're called replication organelles or double membraned vesicles. DMV is the most common thing that people would say. And these are derived from the endoplasmic reticulum. The virus puts some of its own proteins in there, pushes out most of the cell's proteins, and basically just steals the membrane. It's ripping off a hot rod, essentially. And once it does that, it's going to create these little replication organelles or replication factories. Those mean the same thing. Inside the replication factories, we're going to bring in nucleotides, and our genomic RNA is going to come in, and then it's going to be copied inside, and then new copies of the genome and new copies of the subgenomic messenger RNAs are going to come out. The messenger RNAs will be translated. They'll make lots of virus proteins. Some of the genome will be translated. That'll make more DMVs. And the rest of the genome will be packaged up and it will bud. But instead of budding at the cell surface like HIV did, we're going to bud into the ER Golgi intermediate compartment, which is a series of kind of loosely organized vesicles that are located between the endoplasmic reticulum and the Golgi apparatus. So you'll see these viruses sort of budding into what looks like something right next to the Golgi, but it doesn't really look like one of those sort of pancake-like Golgi stacks. And then you'll see the viruses travel through the Golgi, so the Golgi totally fills up with viruses, although that's not where they start out usually. And then it will move through the Golgi stacks, and the Golgi will actually function to bring it up to the surface, which is what the Golgi is for. It's for delivering proteins to the surface, whether you're going to release them from the cell or whether they're like surface membrane proteins. They're going to be part of the plasma membrane. That's what the Golgi is all about. It's like a little bus station. Yeah. All right. So that's our big picture. And here's the big picture in plenty of words. The proteins that we translate are going to be in two groups, and we'll get to their names in just a moment. That's the next thing we'll do. Okay, so here they are. There are two main regions of the first gene. So the first gene is open reading frame 1, and open reading frame means it's basically a gene. It has a start codon, it has a stop codon, and in between those, you could translate a protein. So this is a very cautious way to describe a new sort of sequence that you find. You'd say, well, it has an open reading frame. And then you're not saying it actually gets translated to make a protein, but you're hinting and winking and pointing at it and saying, oh, probably. In this case, though, we know that they are translated, but they've retained the original names. So open reading frame 1A is going to encode 11 different proteins. Open reading frame 1B is also encoded by that gene 1, effectively, and that's going to include another 5 open reading frames that make proteins. And so each of these proteins, I think this is a nice little reference list. We're not going to cover it in detail right now, but this is going to be a good thing to go back to when you are preparing exam answers for this, because this is just like a little key. It's like a little Rosetta Stone that's got a lot of stuff on it. Yeah. Broadly speaking, if we look at these, NSP 1, 2, 3, those are mainly there to shut down some component of the host immune system. They're basically paving the way for the virus. 3, 4, 5, 6, those are making these replication factories. So think of it as like first we clear the way, that's like 1, 2, 3, and then 3 again, because it's a giant protein with lots of functions. 3, 4, and really just 3, 4, and 6, but 5 participates by cutting between those. That's going to make the pores and also staple the two membranes together to make the double membrane vesicle. NSP4 does that. After that, we've got 7, 8, 9, 10. Those are all accessories. They're all going to bind somewhere around the polymerase, and they're going to do important jobs. Each of these has a role to play, but if we were to point out the weak link in the bunch, it would be NSP7. It's just kind of like a brick. It's there between a couple of other things, and it holds those things together, but other than that, it doesn't do anything. NSP9, super important. NSP8, probably an enzyme. NSP10 is like a key that turns on multiple other enzymes and connects them to the rest of the proteins. So yeah. NSP11 isn't really that important. So NSP11 is the piece of protein that coincidentally is sitting on top of a very important RNA signal, which is called the frame shift signal, because this virus does a ribosomal frame shift, just like HIV. Yeah. You remember how we did GAG and we would translate up to a stop codon? And then if we had a pseudonaut and we had a slippery site, we could sort of push back one nucleotide and continue to translate. And that's exactly what coronavirus does. So if it goes up to the stop codon, the little bit after NSP10 is called NSP11. It's tiny. As far as we know, it has no function, but that's where the pseudonaut is. That's the frame shift signal. So I think the RNA that makes that is important, and as far as we can tell, the protein is not particularly important. But if we do frame shift, boom, then we start to make all the enzymes. So just like in HIV, all the enzymes are in that second part, and we only get to them after the ribosomal frame shift. So then the hits start coming. So the first one, NSP12, is the main polymerase enzyme. It reads RNA and it writes RNA, so we call it an RNA dependent, that means RNA reading, RNA polymerase, which means RNA writer. Yeah, basically, yeah. NSP13 is a helicase that actually binds to the polymerase and is essential for polymerase function. These two enzymes being together, this is the hallmark of what makes a coronavirus. Like all the ones that are related to coronaviruses have these two things, and it's a little bit weird. It's their thing. A lot of other viruses will have a helicase, but it'll be somewhere else, and it won't be as integrated as the coronavirus one is, or not in the same way. All right. Then NSP 14, 15, 16, or rather 14 and 16 are going to be capping enzymes, although 14 also is a proofreading enzyme. So this is a very large genome, and the virus actually is trying to control how many mistakes it makes. So the polymerase will make the mistakes, but then NSP14 is like the counterbalance, the check and balance on this. And so NSP14 will correct the mistakes if there are too many mistakes, so that the virus can stay right at its fitness peak and avoid the error threshold. It wants to be real close to that, but not quite over, because if you're over the error threshold, you're just dead. Yeah. All right. Then NSP15 is something we find in coronaviruses of vertebrate animals. So mammals, birds, snakes, reptiles, fish, sharks, stuff like that. They all seem to have and it's an endonuclease, cuts RNA on the inside. And so we think this is an anti-host factor, because when you mess it up, knock it out, the virus can actually copy itself, but it is detected by the cell big time, and the cell will sort of shut things down. So we don't understand it fully, but it's doing something to essentially keep the cell from recognizing that it's infected. Yeah. All right. And so that's it. You got enzymes at the back. You got all your bricks and sort of setup at the front. It's like a mullet. Business at the front, party at the back, with the enzymes being the party. Why not? Yeah. Okay, so now let's go into like, how do these things work? What do these things do? Because that's a whole deal. All right, and so we'll cover only the first part of that in this video, and then we're going to have a video break, and we'll go on to the second part. So, just like we said, there is a -1 programmed ribosomal frame shift just like the words say, which means that the ribosome is going to shift from reading in its particular frame, like it's reading in the frame 1, which is like 1, 2, 3, 1, 2, 3, 1, 2, 3, and it's going to shift it back 1. So it's going to go from frame 1 to frame 3, which is the one before 1. There it is going to continue translation. Doesn't happen all the time, and once again, this is about somewhere between 20% and about 45% of the time it will frame shift. SARS-CoV-2 is actually on the higher end of that. It's, we believe, above 40% effective. And so SARS-CoV-2 has an extra thing. So we have the pseudoknot, which wraps around the ribosome and prevents it from flying apart when it hits that stop codon. And we have the slippery sequence, which is like the little patch of ice. And so we're going to sort of grab our ribosome and slide it back, one nucleotide on the little slippery patch. But then the SARS-CoV-2 sequence actually works too well. It would be too efficient, something way over 50% efficiency. And so there's an extra little loop which forms part of the ribosomal frameshift site. And this is called an attenuator loop, because when the loop is present, then it reduces the amount of frame shifting. So yeah, SARS-CoV-2 has such a good frame shifting device that it actually has to tone it down a little bit. Yeah, which is pretty crazy, right? Yeah. All right, and here's what's going on. So the gray things are ribosomes. We got our large subunit on the top, which is going to do the adding of the amino acids and accepting the tRNAs. We got a small subunit on the bottom, which is going to slide along with the big one and sort of help recognize where we start and stay in frame. And so as we are translating along, one of two things is going to happen. Either you're going to have no frame shift, in which case you just read these codons, just like normal, and ignore that pseudoknot, unwind that pseudoknot, whatever. And then you'll reach the stop codon and it'll stop. And so if we do that, we'll make, instead of making GAG, we say we make polyprotein 1A. So that's polyprotein 1A. Polyprotein is the protein that is made by translating open reading frame 1A. So open reading frame 1A, or ORF1A, is the gene, and polyprotein 1A, or PP1A, is the protein. Yeah, so it's fine. If it doesn't do that, though, it will do this -1 ribosomal frame shift. And here's what's actually going on inside the ribosome. And there are nice structures now that tell us what's going on and sort of back this up. So you'll bind a tRNA in here, and it'll be bound to UUA AAC. And so that's great. And the UUA is going to have a leucine on it, and the AAC is going to have an asparagine amino acid on it, because that's how translation works. That's just the way the tRNAs are. That's why AAC codes for an asparagine. Yeah. Then we're going to run into this, the frameshift signal. And so the pseudoknot's going to wrap around, actually wraps around the back of the ribosome, and it's going to slide it back. The tRNAs stay inside. And so this one, which should be binding UUA, is now binding UUU. And so we've drawn a little line there, but actually that line should be erased because that is a mismatch. That is not a good base pair, because you can't base pair both with A and with U, because, yeah, it's not how nucleotides work. And same thing here. We were binding to a C, so we have a G nucleotide on the tRNA. And we're going to slide that along, so the G nucleotide is now bound to an A, and G and A hate each other, but the pseudoknot is there to keep the ribosome locked down. And basically it's like, I don't know, if you have a dog or something where you have to sort of pinch their jaw and put the pill in there and get them to eat it, Imagine the pseudoknot saying, "This is for your own good," to the ribosome, something like that. All right. And then we have now slid. So we're in a different reading frame. So instead of reading UUA, AAC, GGG, we're now shifted back one. So we're reading UUUAAACGG, like this, UUUAAACGG. And so, yeah, that's how it goes. You put in the next amino acid and carry on. So there will be no stop code on there until we get all the way to the end of 1B. And so if you frame shift at the end of open reading frame 1A, you'll go into open reading frame 2. And if you translate that, you will have translated 1A and then 1B. And so it's not open reading frame 2, it's open reading frame 1B, pardon me. Yeah. And so we call that polyprotein, Polyprotein 1AB, because it's made from open reading frame 1A and 1B with a frame shift. There you go. That's all the magic of naming. So that's plenty for this video. And yeah, I hope you got something out of that. See you on the next one.